as spontaneous induction might occur. So typically, if you just look at the plaques that are formed by bacteriophages, you can see differences between phages which are temperate, meaning those that can form lysogens, as opposed to those that are lytic and are obligated to go through lytic growth. And so temperate phages typically form plaques which appear visibly turbid, as opposed to lytic phages that form plaques that are clear. And the reason why temperate phages form turbid plaques is because within the plaque there's dead cells, there's new viruses that are being made, but there's also those lysogenic cells which are carrying the prophage and are now, they have become immune to reinfection or superinfection by those phage particles. And so a phage that is temperate forms these turbid plaques, and that's an example of a nice turbid plaque there. This is actually another temperate phage which does form lysogens, although it does so at a much lower frequency. So the frequency with which lysogeny is established can vary greatly from different bacteriophage isolate to different bacteriophage isolate. It's normally never more than about 20% of infections, but it can be as low as just a few percent. So the temperate phages form these lysogens. They're genetically stable, and if you take a sterile loop and you scoop some of the cells from the center of this plaque and you streak them out, there are cells that grow. And when you pick these colonies and test them, the vast majority of them are now these lysogens that I've just told you about. So they grow as perfectly happy, normal-looking cells and cultures. They just carry that prophage around with them. And in fact, it's now known from the many hundreds of bacterial genomes that have been sequenced that when those genomes are analyzed carefully, many, if not most of them, carry one, sometimes many, prophages sitting within their genomes. So in fact, lysogeny is not an odd or a peculiar state for a bacterial cell to be in. It is more typical that bacterial genomes carry one or more prophages within them. So I mentioned this property that because there are lysogens that are growing in here that cannot be reinfected or superinfected, they're referred to as being immune. And so what this panel here is showing is the infection of bacteriophage particles on two different types of strains. This first strain here is a strain of bacteria which is a non-lysogen, and then this strain here is lysogenic for this bacteriophage. And this bacteriophage is called Giles, incidentally, and you can see in these spot tests that here we have just a dilution of a bacteriophage lysate, showing single plaques here and more plaques here. But at the same dilution, there are no plaques formed on the lysogen. So this lysogenic strain is immune to superinfection by that particular phage. And you can see a property which is not uncommon, which is that if you have very, very large numbers of bacteriophages in this very concentrated spot, that you do see some killing. There are various explanations and reasons why that might occur. But there is a very large magnitude of effect between what you see at the very high titer, perhaps a millionfold more particles than at the most dilute sample. Immunity, and this property of immunity, is specific. And if two phages are unable to infect the same lysogen, they are referred to as being homoimmune. they have homoimmunity. That's distinct from the state where two bacteriophages can infect the same lysogen, in which case they have different immunity and they have heteroimmunity. So in this example here, these are actually both temperate phages. But the lysogen that we've generated from Giles has immunity to Giles, but no immunity to this phage L5. So this is an example of heteroimmunity. And it's very simple to score for immune patterns by taking a lysogen, as we've done here, and just testing the ability of panels of bacteriophages to infect those two strains. In this particular case, this Giles temperate phage has immunity to itself, but doesn't, so you can see no infection in the lysogen, but plaques here, but none of these other phages share immunity with Giles. So that's an important concept in temperate bacteriophages and lysogeny, is this idea of heteroimmunity and homoimmunity. So I mentioned that lysogens carry a prophage, and in the vast majority of cases, that phage DNA, that prophage, is integrated into the bacterial chromosome. But I should emphasize, and this is a common theme in bacteriophage biology, there's always exceptions. And there are exceptions of bacteriophage genomes that reside as prophages but replicate as extra chromosomal circles. And there are those that replicate as extra chromosomal linear DNA. So there's lots of exceptions. That being said, integration, site-specific insertion of the DNA into the host chromosome is by far the most common event that happens. And I just will explain this in a little bit further detail to say that this is an example, integration is an example of a process known as site-specific recombination, where there are two participating DNA sites. There is one that is a part of the phage chromosome, so a specific DNA sequence in the phage DNA, which is called the attachment site phage, or ATP. And there is a second specific DNA sequence that's within the bacterial chromosome, which is the attachment site bacterium, or ATB. And there is a specialized enzyme, a recombinase, that catalyzes the integration event. And it is imaginally referred to as integrase. Integrase is the name of the class of enzyme that does this. And what I'm showing here is that there's actually two evolutionarily distinct groups of integrases. There are the so-called tyrosine integrases and the so-called serine integrases, where the tyrosine and the serine simply refer to the nature of the catalytic residue which is important for mediating the event. What I want to emphasize is that integration happens at this specific site. Different phages use different sites. And as a consequence of integration, this prophage is now sitting genetically quietly within the chromosome and is flanked by these hybrid sites at L and at R. And so in the process of integration, this integrase enzyme catalyzes this reaction. But I told you that bacteriophages can also be induced or can spontaneously enter lytic growth. And so there is a process of excision that happens where there is another site-specific recombination event, this time using AT-L and AT-R as substrates in order to generate AT-P and AT-B. And the phage DNA can then undergo lytic growth. So if you look in bacterial genomes, I told you many of them carry prophages, and the prophages may look just like this with a full intact bacteriophage genome, but sometimes this process doesn't work well or it's imperfect. You look in bacterial genomes, sometimes you see pieces of this prophage that are left in the genome. So temperate phages typically undergo this process. They don't have to, because the prophages can exist in other ways, but this is quite common. And so when you examine bacteriophage genomes, it's not uncommon to be able to recognize this integrase gene, which catalyzes this reaction, as well as this attachment site.